Subtopic 10.2 Inheritance. Alrighty, so 10.2. Our essential idea is that genes may be linked or unlinked, and then all the traits of inheritance around that. So just in the diagram here, you can see the difference of linked versus unlinked genes. This gene 1 is unlinked from 2, 3 and 4, whereas gene 2, 3 and 4, they're all linked together because they're on the same chromosome, whereas gene 1 is unlinked because it's on a different chromosome. Okay, that's the basics of what we're going to be studying. But then obviously things are not that basic when it comes to biology. So first of all, we're going to have a look at an unlinked situation. Now, unlinked genes segregate independently as a result of meiosis. So let's think what that means. Basically, these genes are always going to be assorted together. That chromosome, unless if crossing over occurs, they're not going to get separated out. Whereas gene 1 could end up separate from gene 2 and 3 depending on how they become assorted during meiosis. We'll look at that in a little more depth, but just let that simplicity of it stay in your mind. So unlinked genes found on different chromosomes, they are unlinked. They segregate independently during meiosis, and it's this term segregation independently that's the buzzword, okay? Linked genes do not segregate independently because they are attached to each other. The only time linked genes might actually come apart is if crossing over occurs. So if you say gene A and gene D and E here before crossing over occurred were linked, well then they're not linked anymore because crossing over occurred. Okay, but gene A and B are not likely to become unlinked due to crossing over. All right. So we're looking at unlinked genes. All right, so we're looking at them being independent. Segregation can occur for genes. Yeah, I've just said that about crossing over. So let's have a look at what we're thinking. This diagram shows it. I prefer this diagram. So each of these genes, these are all showing us individual genes. They will be arranged in all of these different ways. The gametes could have any of these arrangements of genes as a product of meiosis. And that's what it's saying. There's not a specific pattern that unlinked genes are going to follow. They go through independent assortment. Okay. So, a skill. You need to be able to calculate the predicted genotypical and phenotypical ratios of offspring of dihybrid crosses involving unlinked autosomal genes. So we're looking at autosomal, we're not looking at sex-linked. We're looking at dihybrid crosses. So we're talking about two different features we're going to be looking at once. So our cat, I have finally just seen what the differences are. We've got two different cat colours and it's the tail length. So we've got long tails or short tails. Okay, so we've got two different features we're looking at with our cats there. Up until this stage, you've been looking at monohybrid crosses. Okay, so we could have brown hair, blonde hair. Brown hair, blonde hair. Heterozygous cross here, and our outcome from our heterozygous cross is our phenotype is a 3:1. Okay, so we're looking at normal to albino. So albino here is recessive. So we've got 1 albino compared to 3 normal, whatever's we're looking at. Say we're looking at mice. 3 normal mice and 1 albino. Now our genotype, when we look there, we've got 1 homozygous dominant, we've got 1 homozygous recessive, and then we've got 2 heterozygous. So you can see 1, 2, 1. Okay, so that is the expected outcome on a mono hybrid cross. Okay, where we've got 2 heterozygous being crossed. Now, when we come to dihybrid crosses, we're looking at our 2 features. So just now we was looking at tail length and cat colour. Here we're looking at two different fish, and it looks like one of them is also tail length, and I think the other one is eye size maybe. See how they've got big eyes? Okay, so one gene pair there is tail length and one is eye. I think it might mean fin, F for fin, and E for eye. So dihybrid crosses, we're looking at the inheritance of two genes together, and remember your skill is that we're looking at unlinked and independent assortment. So the tail length of the fish has no bearing on the eye size of the fish. Okay, they're independent of each other. Now, Mendel had a look at things like this, and he started sussing some of this stuff out, because he looked at round yellow seed peas. There you go, round and yellow, crossing with a wrinkly green seed peas. So that was his parent generation there. All of the F1s had round yellow seeds. This was due to dominant alleles. So we're saying that round is dominant, we're saying that yellow is dominant, in which case wrinkled is recessive and green is recessive. When Mendel's F1 hybrid self-pollinated, 4 different phenotypes then arose. We had round and yellow, we had round and green, we had wrinkled and yellow, and we had wrinkled and green. Independent assortment, okay, what that means is there is no relationship between the colour and the pea shape. They're not related at all. It is completely independently assorted. So the chance of getting a dominant or a recessive shape gene is half. So 50% chance of getting an R, 50% chance of getting lowercase r. So 50% chance of being round, 50% chance of being wrinkled. The chance of being yellow or green, again, half. 50% for each. So the chance of getting any combination of the two alleles would be the half, multiplied by half, be a quarter. 1 in 4 chance of being yellow and of having a dominant yellow and a dominant green, for example. We're talking alleles, we're talking genotype. So in terms of the genotypical ratio, it's actually a 1 to 1 to 1 to 1. However, in terms of phenotypical ratio, we have a 9 to 3 to 3 to 1. So what am I talking about? Let's have a look. How many round yellows have we got? 1, 2, 3, 4, 5, 6, 7, 8, 9. How many round greens have we got? 1, 2, 3. How many wrinkly yellows? 3. And how many wrinkly greens? 1. Now this ratio is what you will find with any dihybrid cross with independent assortment. Alrighty, so how do we make predictions about our dihybrid crosses? When I say that is, let me come back here. If you are looking at heterozygous crosses, okay, it does obviously depend on your genotype of the F1 that you're crossing, or if you're doing a parent generation of that cross, it depends on the genotype. But how do we actually make the predictions using our Punnett square? First of all, we need to work out the genotypes of the parents. In this case, we again have 2 heterozygous parents. You need to figure out the variety of gametes that can then come from those parents. You then need to set up your Punnett grid, and then you need to work through determine your genotype ratios and your phenotype ratios. Okay, so Mendel did a lot of work a lot of work with his pea plants. Okay, and he got a lot of information. However, science could only take him so far. And then other scientists come along and started looking for discrepancies in his work. And it wasn't a case of proving him wrong. It was a case of building on what he'd found out and advancing science from there. So along came Thomas Hunt, and he was working with fruit flies back in the late 1800s, early 1900s. Okay, and he's working with fruit flies, and he discovered some non-Mendelian ratios in his experiments with his fruit fly. So here we've got a picture of our fruit fly with a red eye, and a lot of his work was around the eye colour, although he also did a lot of work around the wing type. If you can imagine the amount of work that Mendel did with plants, with the pea plants, well, Morgan did all that kind of work with fruit fly, really specifically looking at different features of the fruit fly. Okay, so to look further at some of Morgan's work, we're going to look a bit further at this whole idea of linked versus unlinked genes. Now, we've looked heavily at unlinked genes and independent assortment through Mendel's work there. So now let's have a look at linked genes. So gene loci are said to be linked if they're on the same chromosome. So if genes are on the same chromosome, they're said to be linked. So thinking of your autosomes and your sex chromosomes, if it's autosomal linkage, the genes are on the same autosome, and if it's sex linkage, the genes are on the X chromosome. Okay. So Morgan's work, he actually looked at sex linkage. Okay, bred thousands. He didn't know he was looking at sex linkage to start off with. He bred thousands of fruit fly and observed in those thousands only one with white eyes. So he decided to cross that one with white eyes with a red eye fly. And first generation all had red eyes except for 3 out of 1,200 offspring. Pretty impressive. So a majority had red eyes. However, in the second generation, the white eyes were more prolific, okay? So it actually supported Mendel's 3-to-1 ratio of dominance and recessive. So at this point, Morgan thought, oh, okay. So for some reason, the first cross didn't have that pattern, but the second cross does. So maybe Mendel's work is correct. However, Mendel's work would suggest that there was the 3-to-1 ratio across both males and females. Guess what Morgan found out? All the white-eyed flies were male. So what was going on? Well, the work follows the whole idea of dominance and recessive. Now, basically, the white-eyed gene, there's several genes that are interacting together, but the white-eyed gene on that X chromosome is recessive. Okay, so if you've got a female, you've got several genes interacting together, but if it's female, then due to the recessive nature of the white-eyed gene, the female fly has red eyes. Now with the male, because it only has that white eye gene, then it's white eyes. Now that would be saying in that first batch there were only 3 males, which is not going to be correct, is it? Because if you think about assortment, there should be roughly 50-50, and that's the thing. The white-eyed gene, because it's linked to other genes, only becomes apparent in males, but males can have red eyes as well. Okay, so Mendel was like, well, hang on, what has I discovered here? Okay, because it's shown so much variation within this one fly, or this one type of fly, and he's now thinking, is it just one gene, or is there the interaction of the other genes? And that's why we're going to look now at variation. And I want you to think about a lot of red-haired people, okay? And in red-haired people, they all seem to have very similar traits. They tend to have freckles. They tend to have quite pale skin. We easily sunburn and quite often have green eyes. Is that a coincidence that all those traits are found specifically in people with red hair? Let's have a look further at this whole concept of variation. We've got two different types of variation. Some of these words will remind you of maths. We've got discrete or continuous variation. Now, discrete can also be called discontinuous, just so you know there's different terminologies. However, in IB, we do say discrete, not discontinuous. Okay, so some examples for our continuous. Height, weight, heart rate, finger length, leaf length. So like in maths, they are these measurements as such. Okay, so there's not a distinct category, there's no limit on the value, and it tends to be quantitative. All right. With discrete or discontinuous, these are things like tongue rolling. Now, you either can roll your tongue or you can't roll your tongue. You can't roll your tongue by 1.5. You either roll it or you don't. You know, your fingerprint is different to someone else's. It's not half the same or something like that. They are unique. Okay, so these are distinct categories. There's no in-between values, and they tend to be qualitative. So with continuous variation, there's a lot of gene and environmental impacts, okay? Whereas with discrete variation, there's only a few genes, okay? There's a limited number of phenotypes. Okay, so discrete, our example would be blood groups. That's what we've looked at. It's distinctive. Your blood group is your blood group. You can't measure it and be, I am 25% O today and tomorrow I am 26% O. You're either one blood group or the other. Continuous, this is where things get a bit different because you've got several genes that are affecting a trait. Some genes have like an additive effect. So think about our Morgan's flies, okay? Several genes had an effect on that white eye. Now, Mendel discovered this in his studies where he crossed a white flower to a purple flower, and his F1s, they were purple. He then expected, because he knew his 3-to-1 ratio, so he expected his F2s to have that 3-to-1 ratio. That didn't happen, however. He got a ratio of 1-to-4 to 6-to-4 to 1. What was going on? So he only expected 2 different colours, and he actually got a range of flower colours, as can be seen in the diagram there. So this was due to 2 unlinked genes, and basically they had co-dominant alleles. So the self-pollination of the F1 gave rise to 5 different coloured offspring with a ratio of 1-to-4 to 6-to-4 to 1. Okay, so we've got one white, we've got one really dark purple, and then we go the 4s, and we've got the 6s as well. So if the number of unlinked genes with co-dominant alleles was higher, then you would have even more phenotypical variants. So the more unlinked genes you've got with co-dominant alleles, you get even more variants. Okay, so still looking at these phenotypes of polygenic, so many gene characteristics, tend to show continuous variation. What are we talking about, and why have I put our normal distribution up? The number and frequency of variants can be predicted using alternative rows on our Pascal's triangle. Okay, so if you think about it, that was the row of variants that we saw from our F3s from from Mendel's work. Okay, and then you would move down every other line of Pascal's triangle. As the number of genes increases, the distribution becomes closer and closer to the normal distribution. Okay, so the more genes involved in a specific trait, the more it moves towards normal distribution of expression of that trait. So the closer a trait is to normal distribution, it suggests more than one gene is involved. So if your traits are completely spread all over the place, then potentially it's just one gene that's involved. This we call polygenic inheritance, so many gene inheritance. All right, one of your skills is the identification of recombinants in crosses involving two linked genes. So if you look here in the diagram, we're looking at our linked genes B and C. Okay, so there they're linked, and then when they cross, you get one of each allele from each different chromosome, and that's part of our variation. So recombinants mean genetic shuffling, and you've heard of recombinants when we looked at genetic modification. Okay, so it just means reshuffling the genetics, and we also have recombinants when our chromosomes cross over, so we get recombinant chromosomes. Independent assortment. Okay, alleles of two genes pass on to gametes without influencing each other. So we talked about what independent assortment was earlier. Now, an exception was found when crossing sweet peas with purple flowers and long pollen grains, with those with red flowers and round grains. All the F1s had purple flowers and long grains. What's going on? These F1s were self-fertilized, and then we got 4 phenotypes that were observed: purple and long, red and round, purple and round, red and long. But not in the 9:3 to 3:1 ratio as expected with independent assortment. So people started questioning, hang on, what's going on here? And it is because of crossing over. Crossing over then messed with the ratio. And you can see that here. So you've got your different combinations, okay? So you've got your different section of gene, and how they then, after crossing over here, the different combinations that you would need to get each phenotype. Okay, so it's just showing what's going on. Bit complicated, but just think through what you're seeing. So we've got purple long, so we're saying they are dominant, okay, and we've got our two chromosomes with those genes on, and then so that's the father, that's paternal, this is maternal, so we've got 2 recessive. Okay, so in our gametes we've just got one of them. Then we've got our F1. Okay, so our F1's got both of those genes, all the genes from the parents. Now here we've got their offspring, so we could have that crossing over that's occurred, and that's what causes the variation. Okay, application. Polygenic, so many gene traits, such as human height, may also be influenced by environmental factors. Look, basically what this is saying is, if you have a polygenic trait, such as height, for example, other factors will influence it. So over time, there's been observable differences between height of people in different classes, depending on what job. I'm not talking classes at school, I'm talking social classes, and also depending on what jobs they do and things like that. If you think of 2 twins, identical twins can still have different features due to environmental impacts. Skin colour in humans is partly due to the environment. Sunlight stimulates melanin pigment in the skin, as well as the influence of some genes. Okay, so you do still have these environmental impacts on some of these polygenic traits. We're now going to investigate chi-squared, and how that can be used with dihybrid crosses. Okay, so we're now going to look at how we can use chi-squared with our independent assortment. Okay, so just thinking back to our video on chi-squared, we said we had to record observed outcomes and tabulate in our contingency table with totals. We needed to establish our expected frequency for each outcome. We needed to work out the degrees of freedom using our table, and then we also needed to, so degrees of freedom was using a calculation. We need to use the chi-squared table to establish our critical value or region for P. Okay, and we'll do all this again, but this is just revision from the previous video in topic 4, I believe it is. We had to calculate chi-square for each outcome, add them together, and then compare that to our critical value. We could then decide whether we were supporting or refuting our null hypothesis. So we're going to be looking at chickens. Okay, and we're looking at chi-squared with our dihybrid crosses. So Warren and Hutt in 1936 test crossed a double heterozygote for 2 pairs of alleles in hens. We're looking at the presence of a crest or the absence of a crest. That's the plumage on top of our, can't use the word plumage, that's the crest on top of the chicken's head. Or we're also looking at plumage, which is the colour of the feathers. And we're looking at white, or we're looking at non-white. So, for their F2 cross, so we've had the parents have had offspring. We had our F1s. Okay, we're looking at our F2 cross. There was a total of 754 offspring. 337 of them were white and crested. 337 of them were non-white and non-crested. 34 were non-white and crested, and 46 were white and non-crested. I think I said that all right. So we fill in our observed for each. Now, if you did your table like before, like you were shown in the last video, it would look like this. However, we are assuming independent assortment, and if you use that, it's suggesting that they're actually related somehow. We're looking at independent assortment, so we write it out this way because white crested does not affect white and crested is white and crested. They're independent, okay? They don't influence each other at all. So we write our table out in this manner rather than the manner we learned in our last chi-squared video. So we are assuming independent assortment, which means the ratio that you've learnt for independent assortment, the genotype is 1 to 1 to 1 to 1, and the phenotype should be 9 to 3 to 3 to 1. So our expected ratio, ignore that table, it should be 1 to 1 to 1. So we now need to establish our expected frequencies using the calculation that we've got here. Okay, so our theoretical total is 1. Our ratio total is 4 because it was 1 to 1 to 1 to 1. And multiplied by the total number of outcomes, well, that was 754 because that's how many offspring we had. So our expected frequency is 188.35 per combination. So if we now have a look, so our white crested, we observed 337. The expected outcome was 188.35, and so on for each one. So now we need to work out our degrees of freedom, where we had M as the number of rows, N as the number of columns. So in this case, our degrees of freedom become 3. We now find our critical region for our 5%. So we've got our degrees of freedom as 3, and we're looking at that 0.05 because we're looking at 5%. So our critical region for chi-squared is 7.815. Okay, so now it comes to calculating chi-squared. We need to do this for each of our values, remember. There's our calculation. So we've got our observed values, our expected values, and there I've got the calculations for chi-squared in each of those cells. I then worked that out for each one. Added them all together. Sorry, let's just go back one. Added them all together, which didn't appear in the table. But our total chi-squared was 468.71, a lot higher than what our critical value was. So as the chi-squared is way higher than the critical region, the null hypothesis is rejected. So the null hypothesis was that the traits are not linked and differences between the observed and expected values are due to a sampling error. So we've said that that's going to be rejected. Our alternative hypothesis then was that the traits are linked and the differences between the observed and expected value are not due to a sampling error. So our chi-squared was 468.71. Our critical value was at 7.815. Therefore, our chi-squared is more than our critical value, so we reject that null hypothesis. Thank you for watching 10.2. Hope you've enjoyed it. I will see you again in our next video.